In the video today, we're going to go over the answers to the Unit 9 study guide. So let's start with number 1. Number 1 says molecules that cut DNA at specific places are, remember these are restriction enzymes. They're typically proteins that are able to cleave DNA, and they can cleave DNA that has blunt ends or sticky ends, depending if they want those fragments to be able to stick to other fragments. Number 2. Circular pieces of bacterial DNA that are often used as vectors in genetic engineering are called plasmids. So remember, these are small pieces of DNA that are in addition to the chromosome found in the bacterial cells, and those plasmids can be moved from one bacteria cell to another, and they can also be edited and then inserted into bacteria cells. Number 3. Blank is a process that produces many identical cells. Remember, when cells are identical to each other and they have been copied to become more populated, that's cloning. So when you make a genetically identical copy of something, that is a clone. Number 4. Blank is formed by joining DNA from two different sources. So this is recombinant DNA. When you take DNA from two different organisms and then create one new strand of DNA, it is recombinant because you're combining two different organisms. Number 5. An organism that contains a gene from another organism is called transgenic or transgenic. So this is when you take a gene from one organism and add it to another organism by creating recombinant DNA, and it allows a gene from one species to be expressed in a different species. So examples would be like adding a fluorescent gene from a jellyfish into a rabbit or a mouse or a cat, or adding the human gene for insulin to bacteria so that the bacteria can produce insulin. Number 6, blank joins two pieces of DNA together. So when you have to stick together two pieces of DNA to make one piece of DNA, this is facilitated by an enzyme called ligase. And the word ligate means to tie together. So ligase is an enzyme that ties things together. Number 7. What process can couples use to find out if they are carrying recessive alleles for a disease? And this can be achieved through DNA fingerprinting. If there's couples who are worried that they carry a recessive gene that could be harmful to their children, like Tay-Sachs disease or cystic fibrosis, they might want to know if they're carriers of those disease-causing genes before they start having children. Number 8. Cohen and Boyer transferred a gene from a frog into and this is a bacterium. So this was the first time a gene had been transferred from one species to another. Number 9. A blank is a piece of radioactive RNA that is used to confirm the presence of a cloned gene. And that is a probe. They're oftentimes, again, made of RNA, and they're able to bind to specific genes because the RNA and the DNA that it's looking for have complementary sequences. Finally, on this page, number 10 says what animal was the first to be cloned. And if you remember, this was a sheep, and her name was Dolly. On page 2, we're going to review some vocabulary through matching. So number 11 says SCNT. That stands for somatic cell nuclear transfer. And this is a technique that removes the nucleus from an egg cell and replaces that nucleus with the nucleus of a different body cell or somatic cell. So this is the first step in cloning. So this one, number 11, is E. Number 12 is a vector. And vectors are things that are used to carry DNA fragments into a cell. And so if you have a gene you would like to insert into a plant cell, for example, you could use a virus as a vector to deliver DNA to that cell. So number 12 is D. Number 13 is gel electrophoresis. So this is a technique that separates molecules by using electricity. So electricity is used to run a current through a gel, and that gel sorts things by size while they're being carried by the electricity. And so You can separate fragments of DNA, for example, by size to generate a DNA fingerprint. So number 13 is B. Number 14, T sub I, is a plasmid that is used to genetically engineer corn. So it's a segment of DNA that can deliver a specific gene to a corn plant. So 14 is A. And 15 is a southern blot test. This is a technique that uses radioactively labeled DNA, and it identifies specific genes. So you're able to run an electrophoresis first, and then you can test the different DNAs that have separated out through that test and find out if a specific gene that you need is in that sample. So 15 is C. The next matching set starts with number 16, which is insulin. Insulin is a genetically engineered protein that's used to treat diabetes. So remember, insulin is the molecule that our bodies use to facilitate sugar transfer from our bloodstream into our cells. And without that protein, we are unable to survive. So insulin is really important, and we now can use bacteria to produce it for us. Number 17 is BST, and this is a hormone that is given to cattle, and it increases their milk production. So it's a way for humans to facilitate additional milk production in cows. So 17 is E. Number 18 says hepatitis B, and this is a vaccine that humans were able to derive by genetic engineering. So number 18 is B. Number 19 is factor 8. So if you recall from our notes, factor 8 is a clotting protein that helps blood to clot normally, and it's something that is missing for a lot of people who have hemophilia. So number 19 is D. It's a protein that is used to treat hemophilia. Number 20 is a restriction enzyme. Restriction enzymes create DNA that has sticky ends, meaning that it leaves some DNA bases exposed so that they will stick to other fragments that have the complementary bases exposed. So number 20 is A. The next set of questions is to number the steps of a genetic engineering experiment from 1, which is the beginning, to 4, which is the end. So if you were going to run an experiment that has these 4 steps, the very first step that you would do is to cut DNA with a restriction enzyme. And that's so that you can take a DNA gene that you cut out of a chromosome with a restriction enzyme and insert it somewhere else. The second step would be to take your DNA fragment and recombine it with different DNA from a different organism. So 23 is number 2. Then you're going to clone the cells with the recombinant DNA to find out to see if it's going to be expressed. And then the last step would be number 22, and that's to screen the cells that you've cloned for the gene that you inserted to ensure that your experiment worked. In the next experiment, which is steps 25 through 28, we're going to insert an insulin protein gene into bacteria. So the very first step of that process is to cut out the insulin gene from DNA. So first we need to obtain that gene from the DNA where it's contained. Then you're going to insert that gene into a plasmid. This is so that you can transfer it. The third step is to transform bacteria with that recombinant plasmid. So you're going to introduce that plasmid to bacteria cells so that they can go through transformation, which is acquiring additional DNA. And then the last step would be to extract insulin from the bacteria culture. So once the bacteria have multiplied and have produced insulin, you can extract that and then use it as a medicine. Next, we'll go over some of the multiple choice questions. So number 29 says fragments of DNA with complementary sticky ends. The thing that they can do is join with each other. So the answer to this is C. And it's because they have complementary DNA, which is attracted to each other. They can form hydrogen bonds with each other, and then ligase can tie them together with covalent bonds. Number 30, the use of genetic engineering to transfer human genes into bacteria allows those bacteria to produce human proteins. So this is exactly what we did when we inserted the insulin gene into a plasmid in a bacteria cell. It allows us to take a gene that's important for humans, have the product produced by bacteria, and then we can use the resulting insulin to help people who have diabetes. Number 31. A gene that codes for resistance to an herbicide has been added to a plant. These plants will, and the answer is A, survive when the herbicide is sprayed on the farm field. So this is important because then we can control the weeds in a farm field while still growing the crop without damaging it. Number 32. Bacteria have been transformed with a plasmid that contains a gene for resistance to penicillin. Penicillin is an antibiotic. What happens to the bacteria when the culture is treated with penicillin? When bacteria have bacterial resistance, they will actually live even though they've been treated with an antibiotic. And so bacteria that are resistant to antibiotics can actually be harmful to humans because they are harder to treat with medicine. Finally, number 33 says, which piece of DNA would move fastest in gel electrophoresis? So remember, gel electrophoresis moves things with electricity across a gel. When DNA travels through that gel, larger pieces move more slowly, smaller pieces of DNA move more quickly. So that means that the answer to this question is A, because a segment of DNA that is 50 base pairs long is smaller and is less hindered by the gel than a larger DNA segment, which is 200 base pairs long. So 50 base pairs moves more quickly than 200 base pairs. Next, we're going to review some of the different problems that can be experienced when we are trying to clone things. So problems include advanced aging, tumors, infections, a weakened immune system. Animals can have large offspring syndrome, so they're fatally oversized. Cloning can be a slow process because you have to try many times before it's successful. Organisms that are cloned can also have cardio and respiratory problems. There can be placental abnormalities. So there are a lot of issues with cloning. Despite that, it does work sometimes. Number 35. We're going to list some benefits of genetic engineering in crops. And then some of the things that have been helpful is that we can engineer plants to be insect resistant. We can engineer plants to be herbicide resistant. We can engineer plants that have an increased ability to fix nitrogen, which means that we can put less fertilizer on fields. Plants can also be improved to have higher yields or to use less water. We can also improve livestock by improving the quality of the animals that we're breeding, whether it's for food or anything like that. So there are definitely some benefits to genetically engineering crops. Number 36. We're going to refer to these pictures, and it says, which DNA sequence will be cleaved or cut by the enzyme HPYBI, which cuts at this DNA sequence. We're looking for it to say GTAC on the top and CAGT on the bottom. So we need to look for that specific sequence. And if you'll notice, you'll find GTAC and CATG here in letter B. So this enzyme searches for that specific DNA sequence and then cuts the DNA strand at that location. So the answer here is B. Number 37. We're going to look at this DNA fingerprint and determine who the parents are of a soldier who appears to have been lost in battle. So first, let's look at the soldier. Let's figure out which fingerprint bars line up with different parents. So we have soldier and this letter D match up, and then we have the soldier and this one in letter D match up, and then here again we have a match and a match and a match, and then we can figure out which other bars match. So here we have a match, here we have a match, this one matches, this one matches, and this one matches. So the parents of that soldier are parent set B, and parents C and D are the parents of that soldier. The next one, number 38, says refer to this picture and determine what is happening in this picture. So you'll notice that you have DNA and you have these sequences that are recognized by enzymes. And then you'll notice that this specific sequence appears to be cut here and here. And then we end up with these DNA fragments, and those fragments have some exposed bases, which we refer to as sticky ends. So in this picture, this DNA is being cut by a restriction enzyme at a restriction site into DNA restriction fragments. Finally, on page 6, we've reached number 39 in our study guide. It says to refer to this picture and identify the picture. So what is this picture of? You'll notice it is a gel electrophoresis test of DNA that has separated a bunch of DNA fragments by their size, which means that this is a picture of a DNA fingerprint. And then the last question, number 40, says which enzyme or enzymes would be best for cutting DNA fragments to make fragments with sticky ends? Remember that sticky ends mean that some of the bases are exposed when the DNA fragments are cut. So in this instance, this fragment cuts sticky ends, and so does this one. So the enzymes that cut those fragments are the EcoRI and the BAM1. The HAE3 makes blunt ends, so it would not be a good enzyme for making fragments with sticky ends because it cuts in a straight line between the two fragments that you're creating.